everyone. Thank you for showing up here. I think some of you probably already see me once or twice during this meeting, so sorry about multiple appearance, but I try to make every talk slightly different, so there's something to be expecting, something new here. Yeah, as Wenqi said, STOmics is a company that we focus on spatial technologies. And this is an assay that we'll provide to researchers. They can do whatever research they want on this platform. And I'm going to talk a little bit about what this assay is, what it can do compared to maybe some of the existing technologies out there. And most importantly, talk about some science that's come out of this technology. So I guess since you guys show up here, I don't really need to talk too much about this revolution, sort of evolution of the sequencing, RNA sequencing, right? So way back when the NGS just started, everybody's doing what we call today bulk RNA. So you grind up your tissue, you get an average sort of expression profile. And then when you have the single cell analysis, the single cell platform ready, you can see the transcript of each single cell in the tissue. So you get a variety of transcripts. You do this very nice clusters. However, once you do that, the thing that you are missing is the spatial information among these cells. Now you know the cells, but you don't know where they are in the tissue. So spatial technology is going to advance this one more dimension. I would like to think of one more dimension, that you will have the single cell expression plus the spatial information that you will map these very nice cluster to where your tissues are, right? Exactly where they are. So that's very nice. And I have talked to many researchers here. Some of you guys asked me what's sort of the bucket of different technology. So we would like to think there's two main technology bucket. That one of them is what we call the image-based technology. So those are the ones that you read off an image from a microscope, right? And then the spatial information naturally exists in the image because it just the information there. Another huge bucket of the spatial technology is what we call the sequencing-based technology. So in that bucket, basically you have some sort of a surface, and you put your tissue, very thin sliced tissue on top of this kind of surface. There is some way to capture your RNA in situ, and there is some way to barcode the location so that once you get your library, put it through NGS platform, you will be able to map back into the physical location in your tissue. So for this second type of technologies, usually there are a few things that are very important for this kind of technology when you are considering doing this type of experiment. One is how big the surface is, right? Some of you probably don't care too much because your sample is small, but some people, especially when they're doing large animal primate brain, for example, they need a relatively large surface that can take the whole piece of tissue. Of course, you can piece together smaller ones, but that's tedious and not accurate, usually. So that's one thing. Another very, very important thing for spatial specifically is the resolution, right? We all take pictures with our phones. We know resolution is very important. So that's determined by the size of the feature on your capture feature on your chip, right? The bigger the feature is, the lower resolution it is. So that's another thing that's very important. The third one is how much you capture, and how much you capture, right? I know this conference, most people are interested in animals, which is probably not human and mouse, and plants. The image-based technology, usually they will have a targeted panel, which is limited to certain genes in certain species. Usually commercialized technology will limited to human mouse because that's most market is. For our technology, Stereo-seq, because it is a capture-based, and right now the fresh frozen version will capture the poly A tail. So as long as your transcript will have a poly A tail, we will be able to capture them on our chip. So because of the mechanism, it is unbiased. So you don't need to know ahead of time what gene you want to look at. And it is species agnostic. That means as long as you have a poly A, you're going to be able to do it, no matter it's a plant, it's an animal that's not mouse or human. I actually will show some of these examples of non-mouse and non-human applications. And that's that. And also, we also have some multi-omics capability with this transcriptome. So you can do a little bit of protein profiling on top of that. I'm not going to talk about that in this talk, but we have some products that do that. So specifically to the resolution work, I really like this. This is one of the plot from one of our publications. So here, what we show, what is the resolution of the Stereo-seq is our center-to-center distance is half a micron. So a typical cell is about 10 micron in diameter. So if you do the simple math, you'll see roughly there will be a 20 by 20 matrix on the one cell. And that's 400 cells for each, 400 unique spots for each cell, which is super, super high resolution. For a 1-by-1 chip, I will show later, 1 centimeter by 1 centimeter chip will have about 400 million spots on each of these chips, compared to some of the existing technologies. They have a few thousand features. We have 400 million on the 1-by-1. I love this because it actually showed this in the middle. This cloud is a nuclei image, and you can see these little black dots are our unique features on the chip. You can see there are indeed several hundred of them on each cell. So, and this is a picture of different chip sizes. We have a lot of flexibility in terms of chip size. Our smallest is 5 millimeter by 5 millimeter. Our most popular was 1 centimeter by 1 centimeter. We can go up to pretty big size, but at that point, it's really other accessories like your microscope, your cross-sectioning machine will become the bottleneck. The bottleneck is not really on our chip. So if you really want to like super, super big ones, you're welcome to talk to us, specifically customized chips. So a little bit dig into how we can do that, right? So how can we achieve such a high resolution? This is actually, again, similarly to the sequencer that CG is using, we are using DNBSEQ technology. So this is sort of a repurpose of the sequencing chip. On the surface, capturing surface, there's only DNA balls. There's no physical structure like a bead or well or something like that, which makes this possible to have nanometer size of unique structures. And for each one of these, you will have a barcode for location and UMIs and the capture poly-T capturing. So for each spot, because it has multiple UMIs, it will be able to capture different transcript on each spot. So this is a very high-level workflow. I don't want to go too much deep into it, but one thing I want to emphasize is that this first two boxes are the wet lab work you're going to do. In your wet lab, you do the tissue embedding, slicing, and capturing reverse transcription. There is no capital equipment needed for this part unless you really want to do high throughput. So any individual lab could buy a chip, a kit with chip and reagent, and you can start doing your experiment on the way. So there is no equipment you need to buy, which is quite different from image-based technologies. They always need to buy some kind of a big box because you need to capture the image. And then once the cDNA library is collected, purified and amplified, it's pretty standard NGS library workflow, and you put on a CG instrument, either G400 or T7, do the sequencing, and then it's what come out of the CG sequencing will be a FASTQ file. It's exact the same format you would get from other platform like Illumina. So whatever your NGS software you are currently using can be seamlessly plugged into that. But we do provide end-to-end software solutions for the Stereo-seq data. And we have the image studio, which is basically deal with the image, process the imaging, and the SAW pipeline is our backbone analysis. It deals from like secondary analysis, mapping to a reference genome, to annotation of these transcript, their itself, how much expression, and their locations, XY locations on the chip, as well as their cell ID, which I will talk about that later. And then StereoMap is desktop visualization tool, which is very cool. You can look into the data, exactly zoom in, zoom out, pick up your favorite cluster or favorite gene sets, all that, play with it. The wet lab, as I said, it's a 1.5 day process. If you're really aggressive, you can squeeze that into 1 day. No equipment is needed. It's the stuff that's needed for this process. Usually you already have those in your lab, like cross-sectioning, microscope, general microscope, and PCR machines, things like that. And then, as I said, once you get the library, it's very standard NGS workflow. You go through, you use a T7 or G400, get it through, you get NGS data, which is FASTQ, and then we provide the SAW software. You can download it from GitHub. It's completely free, and we also provide some example data sets on a different website, so you can download the website, download the data set, download the software, try it to see how much you like it, play with it even before you even have your experiment down. And this is the third part, is the data visualization tools. We do provide these data visualization tools. Again, they are completely free, open to the public. You can plug in your data onto that. You can look in these beautiful pictures. You can zoom in, zoom out, pick your favorite gene cluster, what have you. And also we have this StereoPy, which is a Python analysis package. It's, again, completely open source and free. It's similar to, probably you familiar, the other software package like Seurat and all that. And it also, you can use this in conjunction with Seurat and other tools, do whatever you like, or reuse your pipeline for other spatial data. So it's very, very convenient and very cool to look at that. One thing I want to also spend a minute on is that the Stereo-seq is a true single cell analysis tool. What does that mean? Is that one thing you have to have for you to be able to do real single cell analysis is your resolution has to be high enough, right? Your feature has to be smaller than a single cell. That's only one thing. The other thing is that you need to be able to identify where cells are in your tissue, right? Of course, you can use sort of this grid approach, which give you roughly the location of the cells, but we prefer to use a more direct way. And you can see where is my pointer? Where? Oh, yeah.
Your right hand.
On your hand.
Oh, this one actually works.
Yeah. The middle.
Oh. So one thing that we do actually in the wet lab side is that we will dye the nuclei and take a microscope image on the same exact tissue you will do your RNA capture. So this image will be perfectly aligned to your RNA transcript results, right? Once you combine these two together, you will know, based on their location of each cell, you will know which transcript are coming from the same cell. So, and this analysis we call the cell beam analysis is included in the SAW, our backbone analysis. So you will automatically get the cell identification assignment to your transcript in addition to the XY location on the chip. So this is very cool. You get a accurate single cell segmentation, right? This is for animal tissues. For plant tissue, actually, it's more true because for plant tissue, our protocol will stain the cell wall. So this is sort of for the animals, it's referring its cell boundary based on the expression distribution as well as the location of the nuclei. But for plant, because the cell wall is stained, you have absolute choose of the boundary of the cell. That's one thing I really like about the plant analysis on Stereo-seq. So that is a very high level of the technology that we use, we develop, and we use. I'm going to talk a little bit about some examples how this technology has been used. So as I mentioned, it's a species-agnostic, unbiased approach. So it basically can be used in a lot of different species. Obviously, we have most of these publications in mouse and human, but we also have other species and as well as plants. So I'm going to talk a little bit about that. These are just some examples of the publication. And this is a mouse embryo atlas. So this is organogenesis. So they basically, the researchers track as time goes, the mouse embryo development on different days. And then you can get sort of instead of 2D, you have 3D, you have this time progress of that. It's very beautiful. I like to show this because it's very beautiful. You can look at different clusters. You can zoom into your favorite tissue or organization, or you can look at the time progress, how genes change their expression. And I guess the Cell Press agree with me. This is very beautiful. So they picked this as the cover story of that journal. Another one I want to talk about is the ex nihilo brain regeneration paper that published in Science less than a year ago. So this one, I know this is a plant and animal meeting, but I just learned before I came here is that in some area in Mexico, ex nihilo actually are used as a source of food. So kind of fit into the farm animal type of animal. So in this experiment, people actually look at the injury recovery of ex nihilo brain, and again, follow the different days past injury and see how these genes, especially around the injury site, how these genes of different groups how their function change, how their expression change, and how they get back to normal, regenerate. So ex nihilo is very important and very interesting to researchers because it has this amazing ability to regenerate pretty much every part of their body. And hopefully it will shed light on some of these things these regeneration process that will help human in someday. So zoom into one of the layers. So this thin layer right there, it's an equivalent of stem cells in mammalian brain. And historically, we know it's a layer based on anatomic information and staining, right? But if you look at this gene expression profile, you see there's three groups of cells that have very distinguished expression pattern, and their function in this injury recovery is quite different. So, and then you can also look at marker genes to study these genes function and things like that. And also the researchers look at these two very different biological process. One is developmental, one is the regeneration, and they find a lot of similarities between the two, similarities between the groups, clusters, and as well as the genes, which is not very, very surprising, but it's very comforting to see these. I'm not going to talk too much about these, but just show we can do this on Drosophila, zebrafish, and all kinds of different animals, farm animals or not. Let's talk about plant. Plant is a little lagging, I think, in terms of using spatial technologies. There are, as you guys are expert, there are specific difficulties using plant to do any type of research, right? For example, sampling is difficult, and its complexity, diversity of plant is very, very high, and you pretty much have to figure out a way to do one over the other, or even different pieces, on the same plant, right? The leaf may be very, very different from the root, etc. So that's one difficulty. And another is, as I mentioned, many existing platforms are designed for mouse and human only, especially if this technology is a targeted technology. It's almost impossible to have some plant, this pre-designed plant one. And if you want to do it from scratch, it's going to be extremely expensive. And data processing, obviously, is a challenge, which is cross the plant, animal, any sort of things, any new things with this huge amount of data is a challenge. And database is somewhat lacking. I know a lot of researchers in neuroscience, they rely a lot on, say, Allen Institute of their Brain Atlas. Such database is not as good in plant research. That's what I know. And then the price tag, of course. However, despite all these difficulties, there are quite some publications in plant in the recent couple of years. And we can see at the bottom, there's different, our customers have tried different plant, Arabidopsis, obviously, peanuts, soybeans, tomatoes, onions. So a lot of these are very important to our life and our diet. So a few papers I want to just sort of talk about applications. One for the plant research. One is the identification of the cell types and even subcell types, as we saw in the Exnato case, right? You identify new subset of cells. And another one is mapping the developmental trajectory of specific cells, cell group, how they mature, differentiate. That's very important to some of the researchers. And then cell-to-cell communication is very, very important and very, very fascinating to people. This is a Arabidopsis paper. I actually talked about it in detail last year in this conference, but I'm just going to have this one slide here, is that I want to compare these two papers. Both of them look at the Arabidopsis leaf, right? On the left side, it's only single-cell RNA sequencing, and which is published earlier. And the right one is using single-cell and as well as Stereo-seq, which is spatial technology, to look at the same thing. Especially interesting to me is that look at this cluster. It's an epidermal cluster. So in a single cell, once you only look at a transcription profile, you will always almost cluster this epidermal cells all together because their expression profile is so extremely similar, right? It's impossible to separate this, although you know there are two layers of the upper and the lower epidermal cells. So if we look at this paper, if you ignore the spatial information, right, you look at this gene expression profile of these two type of cells, it's indeed super intermingled to each other. It's impossible to separate. But if you consider also the location, physical location of these cell types, it gives you a very nice separation of the two cells, and you can do some Go category analysis of that, and it's actually make a lot of sense. Like comparing these upper and lower, the upper one has photosynthesis enriched, right? And the lower one has respond to lack of light. So they do have different, maybe subtle, but different function in this plant. This is a PNAS paper that's also using Stereo-seq technology, and look at the tomatoes. Actually, I didn't know this before, but tomato, like Exnato in the animal world, it is very exhibit a very high regenerative capacity. So this is how they designed the experiment. They follow this callus formation and then the shoot generation. So this process is very interesting. Again, from each chip or each experiment, you'll get a lot of data. For this study, they totally have 28,000 single cell, and with coverage of about 20,000 genes using the spatial technology, and they get 7 clusters with differential spatial distribution and different expression levels, right? And then you can cluster these, look only at the transcript profile and look at the location as well. And for example, these again, it's 2 different epidermal cells. one is close to the air, the other one is close to the media, right? And then using this information expression profile as well as the location, you can see pretty clearly separate these two. You can look into all these gene functions and marker genes, and you can dig out a lot of interesting functional features of these. Another thing that's interesting, I just pick a few things highlighted from this paper. Another thing I think is very interesting is that the light introduced chiremic cells were found to significantly promote the shoot regeneration, which is kind of interesting. Is that why these sort of photosynthesis sensitive cells are promoting this shoot generation? Supposedly they are on the ground, but from this regeneration process, we can see physically they are close to each other. These are surrounded by the other group, and if you inhibit one of these signals with some agent, it actually decreases the shoot regeneration. So this is very important. I don't know why. I'm not a plant researcher, but it's very, very interesting to me. And again, for each experiment, you get tons of data. You can do experiment once and do the analysis forever. Well, hopefully not forever. Hopefully you'll publish something along the way. The second sort of application, I picked this paper. It's Arabidopsis. I talked to a few researchers during this meeting. It seems like where the flower generation is very important for some researchers. And this one, I really want to pull your attention to here. You can see this trajectory of development from one group of cells to sort of differentiate into mature cells. When I give my talk, the shorter version on Saturday, one of the questions, actually not to me, but to a previous speaker, is that the trajectory sort of prediction, how do you know it's real, right? If you only, for that speaker, she was only talking about single cell analysis, right? For single cell analysis, you can see the cluster, which is based on transcripts profile. You can sort of infer if their transcript profile change continuously like this. It's an evidence of actually they differentiate from one group to another. However, it's still kind of, this is just inferring or guessing. But once you have the information for the location, these cells actually located on the same location, right, along the way of development. And also you see this sort of continuous smooth change in the expression level. You have a much more confidence saying this is how the cell are maturing and differentiating along the way. Cell-cell communication and interaction is always fascinating to researchers, right? And not only in the plant, but also in animals. For example, I study oncology for a long time. Like oncology, the cancer cells, how they interact with normal cells, immune cells, that's all very fascinating. So plant is the same thing. There are two group of methods. One is more like this indirect methods. I just randomly pull out a immune paper. So basically, you know something about the interaction between two type of cells, immune cells versus the cancer cells, things like that. You have some idea how they should interact, and then you look at a gene expression profile. You see how they change together. Are they changing in the same sort of phase, or are they changing counter phase? You look at the relationship. You kind of put a lot of complex methodology and AI, what have you, and you will get some conclusion, right? It's inferred conclusion, indirect, but I would rather prefer a more direct method to really see what's going on there. And it's the famous soybean rhizobia, everybody's interested in if you study soybean. And also here I have a peanut paper. For the peanut, I learned fungi infection is a big problem for them. So not only there are papers talking about this soybean rhizobia, and also there are new methods to detect the plant or animal versus their parasites or fungi infection and things like that. And because, again, this is a technology that's species-agnostic, you can detect multiple species transcript at the same time. So physically, they are next to each other. That give you a lot more information and confidence. So this is very fast through for the new insight in plant. We talk about the advancing developmental in both plant and animal, and decoding the plant environmental interaction or animal as well. And it's also spatial transcriptomics can facilitate the metabolism, pathways, analysis, cell-cell interaction. So despite all these difficulties, as we see that recent publications using spatial technology, especially Stereo-seq, are popping up everywhere. So it's very hot. As I worked in the scientific field for so long, is that one of my personal advice is that once there is a new technology popping out, you better publish it fast. The publication bar is relatively low at the beginning. It will go higher as time goes. We will see that in microarray, early days of microarray and JS spatial, single cell spatial. So I hope this is helpful to you to think about applying using the Stereo-seq to in your research, plant or animal. And I wish you can get big publications soon in these high impact journals. Any questions? Yes.
I really enjoy your talk.
Thank you.
So can you do actually in vivo spatial transcriptomics?
In vivo.
In live tissue. Well, I work with plant and fungi, tracking the fungus, get into the host cell, form a hostorium.
All right.
I want to catch the transcriptome of the invaded cell versus the neighboring cell, and also want to get the transcriptome of the hostoria, the fungal hostoria cell. So really, I want to do it in a live sort of tissue spatial transcriptomics. So your technology, capable of doing that?
Right now, we use fresh frozen, right? So it is live, sort of when you cut it off. And if you want to do a time course, you have to do several different ones, harvest at different time points.
Good, yeah. We can put a piece of meat on the microscope. We can actually, because actually I work on powder mildew, which only invades epidermal cell. So it's pretty easy to extract the single cell.
You don't need to extract single cell. It's your tissue. You slice your tissue, put on the chip, and that's you get the real spatial in action, right? You see, great. Yes, and as you said, because as I said, it's species agnostic. As long as your fungi has a poly A, right? Bacteria right now it's not possible because they don't have a poly A, but we are working on that. But the current technology, you can detect multiple species at the same time.
Okay, sounds great.
Yeah. Thank you. Yeah.
For your symbiotic bacteria or fungi, because the cryosection does not go through those membranes of the microorganism, what's your recovery of their transcript? So when you cryosection, you go through the plant cells, for example, right?
Right. So we cut typically for animal cells, we cut typically 10 microns. So that's one cell thick, one layer of cell thick. We can do from 5 to 20, depending on maybe your cell is smaller or bigger. For plant, I'm not that familiar with that, but
like the bacteria cell that is associated with the plant cell, it will not be sectioned. So there is no protocol for permeabilizing those cells to recover those transcripts, right? So
well, bacteria cell, you can't get them because they don't have poly A in the first place. So we are working on something that's non-poly A. It's going to come soon, but theoretically, because even if you cut through, right, you're not going to just perfectly cut between the layers, right? The cells will be exposed, and also we have permeabilization agent, so we'll try to grab RNA onto the chip and hybridize it.
So, for example, recover of mitochondrial DNA,
we can recover mitochondrial DNA. I think actually it is a standard sort of contamination measurement we have, but if you are specifically looking for mitochondrial DNA, Do they actually have poly A? I don't think so. I think mitochondria is more like bacteria. But the capture rate thing is that it really depends on what tissue, because different tissue have very different features. Usually the animal brain is the best tissue we get. For mouse brain, usually, different spatial technology benchmark their mouse brain data. So for mouse brain, I would say we can get a few hundred genes per cell, sometimes high hundreds per cell. But plant or different tissues, sometimes, some tissue, for example, pancreatic, they have a lot of enzyme and things like that. They will reduce, and also native biology, right? So, for example, the embryo is always good because they express a lot of RNAs. Cancer cells, they express a lot of RNA. You'll naturally capture more. It's really depend on it changes across different tissues. We can work with you on optimization your tissue, especially plant. We really love you guys to have the kit in your hand and do the experiment, because you know your tissue the most, right? You are the expert on that, and we can provide technical support for that, and help you to optimize that for your tissue and your different species as well as the different tissues.
Any more questions even for the previous speakers that are still here, Dr. Henry and Dr. Lee? And Brian, I would love it. If not, thank you very much for coming to our workshop.
Thank you.